Hello everyone, welcome back to my new video. So today we will be discussing about the topic blood indices and packed blood cell volume. So without any delay, let's dig into the topic. Firstly, starting with the definition, what does the packed cell volume means? So let's read that. PCV or the packed cell volume is the proportion of the blood occupied by RBCs and it is expressed in percentage. It is also called hematocrit value or erythrocyte volume fraction. So what is the meaning of this entire thing? For example, as mentioned that it is the proportion of the blood occupied by the RBC. So let's understand it in a simple way. For example, if we take this a shape of a cube and if it is filled with blood, so in this region, and let's say the size of this cube is around 1 into 1 centimeter, the area. So how much in this area, and if it is filled with blood, that means the volume of the blood is also same, that is 1 cubic meter. And in this 1 cubic meter, how much of RBCs are present? Because in blood there are not only RBCs, there are also WBC, platelets and other things. So thus PCV or the packed cell volume means how much volume of the blood is being occupied by the RBCs. And also it is also known as the hematocrit value. Now why so? Because in certain tests the blood is drawn into the Windrom's tube and it is then centrifuged to a very high speed. After that, what actually happens is that let's consider it to be a Windrom's tube. After that centrifugation, firstly at the bottom there will be the red colored cell, which will be the mainly the RBCs, and in the middle next we will be having a white buffy coat, and then on the top we'll be having a yellow colored straw fluid which will be having the plasma. So and it is also used in it is carried in the hematocrit tubes and all the Windrom's tubes, so the name hematocrit value, and other name it is also known as erythrocyte volume fraction. Next, coming to the normal value of the PCV. So the normal value of PCV of the packed cell volume varies from, in males it is 40 to 45%, and in females it is 38 to the 42%. These are the normal ranges. Any deviation from these ranges will lead to pathological or other physiological, abnormal physiological or pathological conditions. Next, the variation in the PCV or the packed cell volume. So the variations mean either there will be increase or decrease from its normal ranges. So first study the causes of the increases. So the packed cell volume increases in condition such as the polycythemia, dehydration and the dengue shock syndrome, that is the dengue fever. And in conditions where it decreases, that is the anemia, cirrhosis of liver, pregnancy, hemorrhage due to ectopic pregnancy and other reasons as well. These are the most common one. Next, let's study what are blood indices. Now, blood indices are the calculations derived from the RBC count, hemoglobin content of blood and PCV. So, as we know that the RBC is a biconcave shell which has no nuclei and it has hemoglobin and the iron pigment attached to it. So it is the blood indices are very important from the clinically and the pathological stand of view because it helps to assess and diagnose many features of the diseases, especially in case of anemia. So let's study them in detail. What are the blood indices or what are the components of blood indices? As mentioned, the main are the MCV, which is known as the mean corpuscular volume, the MCH, the mean corpuscular hemoglobin, and the MCHC, that is the mean corpuscular hemoglobin concentration, and lastly but not the least, the color index, that is represented as CI. So today in this generation, mostly these three are being performed and are clinically more significant. The CI though are performed but on a relatively lower rate than that to MCV, MCH and MCHC. These are very thoroughly performed. So starting with them, let's understand each of them in detail, starting with the mean corpuscular volume or the MCV. So what does it mean? MCV is the average volume of single RBC and it is expressed in cubic microns, and the normal MCV count is 90 cubic micron on an average and it ranges from 78 to 90. That means, so as the name suggests, that average volume of a single RBC. So let's definitely there are in a whenever you will be drawing certain amount of blood in a test tube, or the RBCs will not be of a same size or same diameter, that is a 7 picometer. There might be, it might be 6.2 picometer, or the 6.9, 7.1, it is like that. So on an average, it is calculating all the average value of all the RBCs of the in certain cubic volume. It means the average volume of the RBCs in a drawn amount of blood, and it is expressed in cubic microns. Next, the mean corpuscular hemoglobin, that is the MCH. So, MCH is the quantity or the amount of hemoglobin that is present in one RBC. It is expressed in microgram or picogram. That is the PG, and the normal value of MCH is 30 PG, and it ranges from 27 to 32. 30 is the average one. So what is this? Let's understand this in a very easy manner. Let's consider this to be an RBC. Pardon for my drawing. And we know that in the RBC there is the iron containing pigment. So what is actually hemoglobin as well? So one RBC is occupy the hemoglobin, and what actually happens is that how much hemoglobin is, or the quantity of the hemoglobin that is present in one RBCs. So how much hemoglobin, or the quantity of the hemoglobin, the assessment of the quantity of hemoglobin in one RBC is nothing but the MCH, and it is expressed in the picogram or the microgram, and the normal value is ranges from 27 to 32 with average being 30. Next comes to the mean corpuscular hemoglobin concentration, MCHC. Now it is very much important because it helps us to diagnose the anemia very clinically and very precise manner. So MCHC, or the mean corpuscular hemoglobin concentration, is the concentration of hemoglobin in one RBC. It is the amount of hemoglobin expressed in relation to the volume of one RBC. So let's understand it in a very simple manner. As I mentioned that the RBC is a cell that occupies, or that actually houses the hemoglobin in it. So what happens is that the amount of hemoglobin, if it is more, not actually, the fact that we are considering here of a single RBC. So there can be two conditions: either the hemoglobin concentration is of normal value, and or the hemoglobin condition is of lower or lesser value. So in certain cases where the hemoglobin is considered as, or is found to be normal, in such cases it is known as the normochromic, and in the another cases where the hemoglobin concentration is lower, then it is known as the hypochromic. Now how to remember it in a very easy manner, just like I did. Normochromic. Now chromic means generally something that has staining properties or has color. So normochromic, if the hemoglobin count is normal, so that definitely it will be having the normal red color. And if the hemoglobin count is, the concentration is lower in a single volume, in a single RBC, then definitely the lower hemoglobin, that means the lower color, so hypochromic. In this way we can easily remember it. But however, it cannot be hyperchromic. Now why hyperchromic? Because there are certain limitations for the concentration or the housing the hemoglobin concentration. That means a single RBC cannot be hyperchromic. Overall it can be, but not a single conditions. So this is very much useful. So why so? Because let's see, in certain conditions such as in megaloblastic anemia, and the pernicious anemia. So both of them are actually due to the vitamin B12 deficiency, as a result of which there is the cytoplasmic defect, and for a result of which there is the defective formation of the RBCs. And in those conditions, the RBCs are actually normochromic in nature. They are normochromic and microcytes. I will be explaining the term microcytes and the macrocytes now. And also in condition that is in iron deficiency. In conditions that is iron deficiency, the cells are hypochromic and sorry, that in megaloblastic and pernicious anemia, it will be normochromic and the macrocytes. And in iron deficiency, it will be hypochromic, and at the same time, it will be microcytes. So, the simple thing, in megaloblastic anemia, it will be normochromic, and it is generally called as the macrocytic normochromic, and in iron deficiency, it will be called as microcytic hypochromic. So, just because of the fact that in pernicious anemia and in megaloblastic anemia, that is vitamin B12 deficiency, which causes cytoplasmic defect, as a result of which the cell will be smaller, and also there will be normal hemoglobin concentration. So that is why it will be macrocytic, since the larger volume of cell, and also the normal hemoglobin concentration, so normochromic. And iron deficiency, that would be the normal cytoplasmic volume. Actually what happened in iron deficiency is that there are nuclear defects, as a result of which what is happening is that the cells are being dispersed into the main bloodstream at a very early stages, for a result of which they are actually smaller in size. So that is why they are the microcyte. So that in such cases it will be called as microcytic, and lower amount of hemoglobin, that means the lower hemoglobin concentration, so hypochromic. So microcytic hypochromic. And also to the fact that the mean corpuscular volume, now actually it can be of two types, that is the macrocytes and the normocytes and the microcytes. So what are these? Actually, whenever the volume of the RBCs are bigger than the usual, then it is known as the macrocytes, and the normal shape of the RBCs, if it is found, it will be called as a normocyte, and the comparatively lesser volume, it will be called as a microcytes. Now the macrocytic anemia is found in conditions such as vitamin B12 deficiency, and also in pernicious anemia and other cases. Normocytic anemia is found in conditions such as the prolonged bleeding or in accident cases, etc. And microcytic anemia are mainly caused due to the nuclear defect, such as the iron deficiency anemia. So, and at last but not the least, the color index. The color index is the ratio between the percentage of hemoglobin and the percentage of RBCs in the blood. So actually it is comparing the percentage of the hemoglobin and the percentage of RBCs. Actually the percentage of hemoglobin in one individual and to that of the average individual, or an individual that are having a normal count. So these are actually the blood indices that are mainly followed in the pathology lab also in detecting the types of anemia, that is the MCB, the MCH and the MCHC, because it will help you clearly assess the types of anemia, and also it is found in several other tests as well. For example, if the results comes out to be macrocytic, then it is often considered to be due to the vitamin B12 or the folic acid deficiency, or also due to pernicious anemia. If it is normocytic, that is in normal volume, then it will be often due to the prolonged bleeding or due to bleeding conditions. And also if it is of lower or the comparatively lower smaller size, then it will be the microcytes, that is found in conditions such as the iron deficiency, nuclear volume, and also the concentration, that is the MCHC, which is very important because it strictly tells us about the hemoglobin concentration, the iron concentration. That is, if the iron concentration, hemoglobin concentration is normal, then it will be normochromic, such as in vitamin B12 deficiency, and if it is less, that is found in the iron deficiency, then it will be called as the hypochromic. So with this, we come to the end of this topic. If you have any doubt regarding this video, do let me know down in the comment section below. Thank you.